Hi everybody, welcome back. I think you are here after having watched the video where I discussed about the cell membrane. Hi, this is Varsha Kulkarni and I'm from Darwin Academy. Let's continue discussing about osmosis from where we left in the previous video. Alright, so that is why I've written osmosis continued. In the previous video, I have discussed about the definition of diffusion, osmosis, and whatever topics were related to the cell membrane. Okay, I had to talk to you about a few examples of what would happen if a cell is placed into varying or different types of solution, and we would be studying different types of osmosis here. Though for your NCERT curriculum, you need not have to know the types of osmosis, okay? But you should know the varying solutions in which we would place animal cell and what would be their outcome. With that being said, I'll also be discussing about more details of cell wall slightly later. So I don't want you to worry about this part of the board at the moment. Alright, so we will be focusing more on osmosis. Like I've mentioned, osmosis is the movement of water molecules from their high concentration to their low concentration through a semi-permeable membrane. I've also told you that it is called as specialized diffusion as well. Okay, now if I place a cell, okay, into a solution which is dilute. Okay, this dilute solution has more water. Do you agree with me? Dilute solution will have more water, isn't it? So this solution is also called as hypotonic solution. All right. So now I'll be taking the example of dry resins. Dry resins. These are the dried grapes that most of us would find in our kitchen, right? So if I place the dry resin or the dry grapes that you may know of into a solution which has more water, that means it is a dilute solution, what might happen if I allow if I allow the resin to stand in this dilute solution for long, what would happen? There is more water here on the outside, okay, and very less sugar here, okay. I've told you, remember, that this is a dilute solution of what? I've taken water, probably say around 100 ml or 150 ml of water, then I've added in a teaspoon of sugar, stirred it. So this is a dilute sugar solution, isn't it? So if I place these resins, this green color here would indicate the resin for you. If I place it there in the dilute sugar solution, you know that this sugar solution will have more of water molecules than the sugar, isn't it? If I allow this setup to stand for a couple of hours, then what will I observe? After a few hours, the water molecules which were here in the solution would have moved into the cell, and as a result the cell would have swelled up, okay, or it would have become swollen. Sometimes when you're feeling tired or when you feel that you have less energy, probably your mother would say, okay, I'll soak a few dry resins at night and probably in the morning you may consume it. What will happen if you soak these dry resins in water for some hours? The water molecules will start moving into the resins, and in the morning you see that the resins would have swollen. How did this happen? Through the semi-permeable membrane of the resin, the water molecule which was in the beaker would have moved into the resins, making the resins to swell up. Okay? So, here you should know that the solution has more water, right? It has more water, and as a result, water molecules have moved from their high concentration to their low concentration. See, the resins were dry, that means there was very little water here inside. So the water molecules from the beaker have made their way into the resins through the semi-permeable membrane of the resins, and as a result, the resin would have swollen up, right? It would have become swollen. This was one part of it. Now, in another situation, consider that I have taken around the same quantity of water, probably 100 or 150 ml. Then I have added so much sugar, around 6 teaspoons or 5 teaspoons, and stirred it nicely. Okay? And then if I add the dry resins and allow this to stay as such for about 6 to 8 hours or 2 hours or whatever, then what will happen? After those few hours have elapsed, then you'll see that the dry resins would have reduced in their size. Why did this happen? It is because there was the water content within the resin was more than that of the beaker, because in the beaker you have more of sugar and less of water. So this was a concentrated sugar solution, which is also called as hypertonic solution. So as a result, the water molecules from the resins would move out and into the beaker through the semi-permeable membrane. As a consequence, the dry resins, first of all, they would be shriveled up and they will shrink further. Okay? Shrink further when placed in a concentrated solution or a hypertonic solution. Then let us consider another example. If I take again the same amount of water, either 100 or 150 ml of water, and add around 2 teaspoons of sugar or somewhere similar to that, okay, and stir it nicely. Then I place dry resins inside this. At some point, even if I keep them for hours, the dry resins will neither swell up nor will they shrink. Okay? But why? It is because the amount of water inside the cell and outside the cell was the same. And as a result, water molecules neither entered the cell nor did they come out of the resins. Such type of solution is called as isotonic solution, when there is no change observed in the material, that is the cell or the resin that you have added into such solution. Now, there is one more inference that we draw from here. We understand that the concentration of this solution and the concentration of the solution inside, that is the sap of a cell, will be the same, isn't it? So now I've discussed the further continuation of osmosis. We have taken 3 different situations, 3 different beakers where we have varying concentrations of sugar, and they are called as hypo, hyper and isotonic solutions respectively. And we have also studied what will happen if a few dry resins are placed in hypotonic solutions, hypertonic solution and isotonic solution. With that being said, we will draw or we would wind up the session on osmosis, and we will move to the other part of the cell. Now, if I take a plant cell as an example, remember in the last video I told you that it is only the plant cells that are having the cell walls. In this section of the video, we will be learning about what the cell wall is made up of, okay, what is its function, and also an important concept, that is the plasmolysis. Okay, so cell wall is the outermost protective covering within a plant cell. You don't find cell wall in an animal cell. It is the outermost layer in a animal cell only, in a plant cell only. It is made up of cellulose. Cellulose is a complex carbohydrate. And remember, this wall or this part of the cell, that is the cell wall, is dead. It is not living. Okay? Whereas all the components inside from the cell membrane onwards are living. Okay. See, but why should something that is dead be present within a plant? Let's move to the function to learn about that. See, there is lot of water being taken in through the into the plant cells, and also the water is lost through the plant cells by transpiration, isn't it? So this movement of water into and out of the plant cell is more, right, when compared to the animal cell. That is the reason why the cell wall is present. There is a lot of changes happening every now and then in terms of the pressure inside the animal, inside the plant cell. So to withstand that pressure, the cell wall is very strong. It is made up of cellulose, and it is a thick layer, and it is dead, and it protects the cell from bursting. Okay. If water keeps continuously entering and leaving the cell, then there is a lot of pressure changes. The cell has to be able to handle these changes. That's the reason it is being provided an external layer, which is the cell wall. And it is very thick. It provides mechanical support as well to the plant, and also a shape to the plant cell. You remember that we as animals have the skeletal system, but plants don't have the skeletal system. There are to be such special layers that provide them with the mechanical support, protection, and also which will provide the shape to the plant cell. Okay, so the reason the cell wall is present and it is dead is because there is too much of osmotic changes happening when water enters or leaves the cell. Every time there is change in the pressure inside the plant cell. In order to withstand that, the cell of a plant is being provided with the cell wall. Okay, and also let's move to the last concept that I would like to discuss before winding up the session, and it is important. It is the plasmolysis. Split the word here, plasmo and lysis. Plasmo refers to the plasma membrane. Remember, we have discussed about the plasma membrane, that is the cell membrane in our previous video, and lysis meaning to break or to tear. This word simply implies that there is tearing away of the plasma membrane. But why? But how? See, if I place a plant cell into a concentrated solution, the water molecules from the plant cell will start moving out into the solution, right? So much so that the cell membrane, look at this diagram here. Initially, the cell membrane is very close to the cell wall. If I place this cell into a hypertonic solution for a long time, then the water from this cell is lost. There is so much of water lost from the plant cell that the cell membrane will start tearing away from the cell wall. That diagram I have drawn here. This black outer layer is the cell wall. I have drawn it in two layers just to help you understand that it is a thick layer. Okay. Then I have used the same green marker to denote the cell membrane. See, initially the cell membrane is very close and it is almost sticking to the cell wall. But what has happened here? It has peeled itself away. It has moved away from the cell wall almost towards the center. The cell wall is remaining intact. There is no change in it. But since lot of water from the cell is lost, the plasma membrane has torn away, peeled away from the wall, and it is gradually moving to one side. So this process wherein the plasma membrane has moved away, torn itself away from the cell wall and starts moving to one periphery, this process is called as plasmolysis. Why did plasmolysis occur? Because most of the water molecules from the plant cell have moved into the outer, or they have moved out of the cell. Okay, this will happen when the cell is placed in a hypertonic or a concentrated solution. Okay, so this is extreme case of cell being placed in a hypertonic solution for a longer period of time. Another question, can plasmolysis be reversed? See, now the plasma membrane has moved to one side along with all its contents. Can it be reversed? Can it be brought back to its original shape? Yes, we can, provided we place such solution in a such cell within a dilute solution. So we have discussed about different types of solutions and what would happen to cell when we are placing them in isotonic, hypotonic and hypertonic solutions. Also, we have discussed about cell wall, its composition, function and plasmolysis. With this, I'll be ending the video now. Thank you.